Hello, everyone, and welcome to another episode of Let's Talk Hereditary. We have Dr. James Church. Dr. Church, thank you for being with us today. In this episode, we want to talk a little bit about the history of genetics, inheritance, and some of the things that happen around the United States and the world that are related to that. So let's start with talking a little bit about the word genetics. What does it mean, and where did that all start from?
Well, to understand the word or the concept of genetics, we have to go back in time, but not particularly far back to the last half of the 19th century. And it relates to this Austrian monk, Gregor Mendel, who became interested in inheritance during his studies before he became a monk. And then the monastery that he was in had a big garden, like several acre garden. And he was given permission by the abbot to go and do experiments with the peas that were growing in the garden. And by crossbreeding the peas of various colors, he was able to come up with Mendel's laws of inheritance, which we know today as dominant inheritance, recessive inheritance. Amazing, really amazing. And he published this stuff, and like what happens to many prophets in their own backyard, it was ignored. And then it surfaced again towards the turn of the century. The word genetics came into being really early in the 1900s, and that led to the word gene and genomics a bit later. But Mendel recognized that there were invisible substances or an imperceptible way in which traits were passed on from one generation to another. He knew that there had to be that. He just couldn't conceive of a name for it. He didn't know exactly what it was. We didn't know about DNA then. We didn't know about nucleic acids. He didn't know about the genetic code. He just knew that if you had a yellow pea and a green pea, you had like 50% chance of getting a yellow pea in the offspring plants. And he analyzed that and knew that there was passage of a trait from one generation to another.
So he was a monk, not a scientist initially.
He was a monk. He became a monk because it was a good way of getting his schooling paid for. So he was a monk far ahead of his time. Wow.
So all this genomic science that we know of started in the backyard of a church somewhere.
A big garden of a monastery, please.
Wow. Okay. All right, so we talked about genetics and explains Mendelian inheritance, and you mentioned a little bit about autosomal and dominant genes. So what is the main difference between both? Autosomal and dominant.
Well,
recessive and dominant.
Okay, so a dominant inheritance happens when there is a gene that's transmitted that you only need one copy transmitted to the offspring for the disease or the trait to occur. In other diseases or traits, you need two copies to be inherited, so one from mum and one from dad, and that's recessive. Having just one copy passed on creates the carrier phenotype, which isn't associated with the trait or the disease. So it's essentially the number of copies of the gene that you need to get from a parent or both parents to develop the disease or the trait.
And I mentioned the word autosomal, but there are also something called sex-linked genes. We don't think about it a lot in colorectal cancer, but what is that?
Well, there are 26 chromosomes. There are 24 autosomes, which are non-sex related, and there are the two sex chromosomes, X and Y. And as you said, sex-related inheritance is pretty uncommon, but most of the diseases that we deal with are autosomally inherited.
And what's a mutation? How does this or a normal DNA turn into a problem that's causing a disease right now?
Well, DNA is the molecule of life, essentially, and it's a huge, complicated molecule. It has about 3 billion what we call base pairs. So DNA is essentially a sequence of 4 nucleotide bases: adenine, thymine, guanine, and cytosine. And these are arranged in a very long, actually a spiral configuration, up to at least 1 meter long, that's condensed and stored in the tiny nucleus of every cell in our body. It's really amazing when you think about it. Within the DNA, and strangely enough, only accounting for about 5% of the DNA, are the 20,000 genes that hold the recipe for every protein in our body. They make us who we are. When we think about the three main food groups, fat, protein, and carbohydrate, carbohydrate and fat provide energy and store energy. Protein is function. If we lose protein, we lose function. If we lose function, we can get diseased. That's the importance of DNA coding for every protein in our body. Every time a cell divides, that DNA molecule has to split itself in half. The two strands come apart. One goes into one cell, one goes into the other. And then in those new cells, the daughter cells, new strands are created by DNA polymerase to make a full molecule of DNA for that cell. So with all these billions of base pairs splitting and dividing and reforming, mistakes happen. Now, the importance of having faithful DNA replication is so serious that there are multiple repair mechanisms designed to detect DNA mistakes and to fix them. Sometimes they don't work. And it's interesting that of the syndromes of hereditary colon cancer, most of them involve a failure of a repair mechanism. And that leads to multiple mutations in multiple cells. But mistaken DNA, if it gets passed on to the next generation, becomes permanent and becomes a mutation. So a mutation is a permanent structural change in the DNA that may or may not influence the protein formation. If it's not in a gene, then basically who cares? If it's in a gene and causes no change in the protein, then who cares? Those are called polymorphisms, so different shapes of the DNA, but no functional problem. If it's in a gene and it impairs gene expression, so it changes the protein, then we could be in for trouble. So not all mutations are bad news, but some of them are, and most of the bad news comes from mutations.
So speaking of mutations, we've discussed before something about the origin of colorectal cancer carcinogenesis in the Vogelstein-Ferron paper. So tell me a little bit about the history of the APC mutation.
Well, APC mutation, as we know now, causes familial adenomatous polyposis. And we've known about that syndrome for a long time, for at least the beginning of the 20th century. It was realized that young people had lots of colon polyps and there would be multiple relatives with it. However, we didn't really have much idea or thought about what was the reason for that until we started to find out about DNA. So one of the most important clues that we had to where the gene causing FAP was in a paper published by Lemuel Herrera and others from Roswell Park in 1987, where they described the case of a patient who had polyposis and a deletion of chromosome 5. And that pointed the finger to chromosome 5. All we needed as a community of scientists was that clue, and that started a series of linkage analysis experiments, which again finally ended up showing us the position of the gene on that chromosome. And it wasn't too long after that, so 1987 was Herrera, about 1988, 89 was the linkage. Walter Bodmer was the one that did that. And then 1991, the gene was cloned or identified, and we knew where it was. And then cloning the gene led to tests like protein trungation test, because we knew which protein was involved, we knew how the disease happened, and not only did we find out about familial polyposis, but because it's such a key gene in sporadic colorectal cancer, we found out a lot about that as well.
So you mentioned also a little bit that one of the main syndromes, or most of the syndromes, are due to a failure in the repair mechanism, which is the mismatched repair genes that are causing Lynch syndrome. So can we talk a little bit about the history of Lynch syndrome? It's always intriguing how we moved from Amsterdam criteria to HMPCC to Lynch syndrome.
So Lynch syndrome history goes back to the late 1800s in Michigan, where a professor of pathology called Aldred Walton one day was going to see her seamstress to pick up his suit, and she was sad. And he said, "Why are you so sad?" And she said, "Well, I'm going to die, and I know what I'm going to die of." And he said, "Oh, tell me more." And she said, "Well, my father died of stomach cancer. My aunt died of uterine cancer. I have several relatives that all died of these cancers." And Walton thought, "Oh, I wonder why." And he researched this family, which he called Family G, because they came from Germany, and found this dominant vertical transmission of cancer, colorectal cancer, gastric cancer, and uterine cancer, over multiple generations of the family. And he wrote about it. His paper was published in 1913. And again, just like Mendel and his observations, ignored. Nothing happened. And until the 1950s, when a professor of medical oncology in Creighton University in Omaha, Nebraska, named Henry Lynch, had also been observing in his own patients that cancer seemed to run in some families from generation to generation. Then he found Walton's paper, and in 1971 he published Walton Family G Revisited, which was his take on hereditary colon cancer. And of course, everybody poo-pooed that. He applied for grants to research it, turned out. The next significant thing was a medical student got interested, named Rick Boland. And Rick wrote really a fabulous paper on the history of Lynch syndrome, which is really worth reading. And he coined the terms Lynch 1 and Lynch 2. And I remember using those terms back in those days when I was a neophyte. So Lynch 1 was colon cancer specific, and Lynch 2 was colon cancer and other organ cancers. We now know it's the same syndrome, and there really isn't a Lynch specific syndrome, but it looked like it was there. And then the name changed, and this was Henry Lynch did this, to HNPCC, because he thought, well, I don't want to have colon cancer family syndrome, which was his original name, that could include FAP. So he wanted a way of excluding polyposis. So he called it hereditary non-polyposis colorectal cancer in 1985. And it stayed like that for decades. And a lot of other scientists got interested in it and set up this international collaborative group on HNPCC. The name didn't really change to Lynch syndrome until 2005, where it became specific for the genetic mutation in the germline. The genes themselves weren't found until 1992 and 93, 92 for MLH1, MSH2, 93 for PMS2, and 97 for MSH6, and then EpCAM came in about 2002. So fairly latecomer genes, but with HNPCC, people were working it out. Along the way, microsatellite instability was discovered back in the 90s, and then immunohistochemistry, looking at the expression of the genes once the genes were known.
So you mentioned an international group for HNPCC. What happened to that group?
That group merged with the group that had been established for familial polyposis, which was called the Leeds-Castle Polyposis Group. That was established in 1985. It's named Leeds-Castle after Leeds Castle in the United States, which, interestingly enough, is not in Leeds at all. It's down in Surrey somewhere. And that happened because one of the doctors at St. Mark's saw a patient with desmoid tumor and didn't know how to deal with it, well, found it difficult to deal with it, so he wanted to get a group of people that were interested in FAP together, and he did. And that was really the first working collaborative group for a inherited colon cancer syndrome. So these two groups existed in parallel for some years, but the same people would go to each meeting. So it was inevitable that they would get the idea to unite the two groups and just have one joint meeting, which happens every 2 years. The united group is called INSIGHT, or International Society for Gastrointestinal Hereditary Tumors.
And I think there's another group in the United States that also focuses on hereditary cancer and genetic syndromes. What is that?
Well, that group is called the Collaborative Group of the Americas for Hereditary Gastrointestinal Cancers. It was established in 1995 with the thought that there are things in the society and in healthcare in the United States and other parts of the American continent that are different to Europe. And so it's also much more easy to travel within the American continents than it is to go gallivanting off all over the countryside in Europe. So for those two reasons, the American group was set up.
And is there any other group in the world that also studies the same syndromes and focuses on them?
There's a European group which was established for European purposes, originally called the Majorca Group, and now called the European Hereditary Tumor Group, meets every year. And there's a South American group, which is called GEF, but I can't really tell you it was established in Brazil, and the words are Portuguese.
All this history talk is very intriguing and interesting to me, but I would like to see what is your version about where the future is heading and what does the future hold for all this collaboration?
Well, we've come a long way. There's no doubt that we've come a long way. And a lot of questions have been answered, but as with anything in knowledge, the more questions you answer, the more you know you don't know. I think we have a lot to learn genetically. I think we need to know what are the modifying factors for gene expression in these syndromes. Not everyone with Lynch has the same risk of anything. So not everyone with Lynch has the same expression of cancer risk or the same manifestations. Even within a family, the expression of the disease can be different. So what are the modifying factors? Where we can refine risk and offer more specific treatment to people? We have lots of issues with colorectal cancer in general, especially young age of onset colorectal cancer. How to deal with this? Why is it happening? Cancer vaccines are just starting. The whole idea of immunotherapy and enhancing the immune system for defense of cancer and curing cancer we just opened the door a crack to that field, and that could be the answer to a lot of our woes. So the future is extremely exciting, but right now we have thousands of patients to deal with. So we need to know better how to deal with patients, how to choose our operations better, how to do them better, how to follow patients better, how to save lives, how to prevent cancers. That's what we're all about at the coalface. And then the scientists in their labs can find out more about the disease and then provide us with the tools to advance clinically.
There's also one group that you're the principal investigator for the Cleveland Clinic. What is that group and what is its focus?
Well, I think the group you're referring to is the Collaborative Family Registries. So years ago, when I was a young man, we got involved in the polyp prevention group, which was a multi-institutional collaborative group doing polyp prevention research. It was headquartered in Dartmouth, and it included several different institutions throughout the country. So the idea was to get patients that were getting polyps on colonoscopy and give them something to stop the polyps, and giving another group, like sugar, and seeing if there was a difference in polyps. And so these were placebo-controlled, prospective, randomized studies. So the first one we did with vitamins A, C, and E, and then we did folate and calcium, and then selenium. And we found that vitamins A, C, and E didn't make any difference. Calcium did. And several other groups have researched the same sort of thing, but the institutions that had all banded together, when that steam ran out of that effort, decided to put together an application to the National Cancer Institute for a big project to gather patients with colon cancer to do research on their lives, their lifestyle, their DNA, and their family history, and see if we could find out something about colon cancer. And really the idea was to create a resource that anybody could use. And so over time we enrolled thousands of patients that had risk factor questionnaires and family history questionnaires. We got their blood, we got their DNA, and all of that's available if you have a question. So the clinic has contributed families with HNPCC and Lynch syndrome to this effort, and there have been hundreds and hundreds of articles written. A lot has been found out because of this wonderful collaborative group, and we really count ourselves fortunate to be a part of it.
Well, thank you for all this information, Dr. Church. Thank you everyone for being with us today. This was a good talk about the history of the disease, the syndromes, where everything started and where the collaboration is heading in the future. If you have any questions or concerns, please feel free to reach out to me or to any members of the Wise Center. Thank you.